to your ST 210 Week 6 mini session, Chapter 14, Actinobacteria. Let's get into our chapter objectives. Now, by the end of this chapter, you're going to be looking at key terms. We're going to compare and contrast the different genera of Actinobacteria. We are going to describe Corynebacterium pathogens and the diseases they cause, describe the Mycobacterium pathogen and the diseases they cause. We're going to look at implications of drug resistance in tuberculosis and apply critical thinking skills in relating all the information in this chapter to our environment of care on a broader global community. But understanding streptomyces and its importance in medicine is extremely important. Streptomyces is a type of bacteria that plays a critical role in the pharmaceutical industry, especially in creating medicines for people who are sick. It's particularly helpful for treating infections caused by bacteria, fungi, and parasites. Not only that, but researchers have also discovered that some products from streptomyces can help fight cancer. As an ST student, you're going to likely encounter several important antibiotics that come from streptomyces, such as streptomycin, tetracycline, neomycin, just to name a few. There are also antifungal medications like nystatin that are derived from the same family of bacteria. Food for thought. While streptomyces is known for its medicinal benefits, it can sometimes be confused with other bacteria and has the potential to cause infections, just like many other bacteria do. Researchers study how streptomyces interacts with other bacterial strains to learn how to fight infections more effectively. They look at the ways that streptomyces defends itself against other bacteria to help develop better treatments for patients. So understanding streptomyces is important for anyone in our field, especially surgical techs, because it helps in both the development of life-saving medications and ongoing battle against infections. All right, that was our clinical significance topic. Now, we're looking at Actinobacteria. This is a class of bacteria that are gram-positive and have a high G+C content in their DNA. Now, this group is diverse, meaning that the bacteria can come in various shapes and sizes, and they have different lifestyles. Some of these bacteria thrive in oxygen-rich environments that are aerobic, while others can survive with or without oxygen. One of the most important roles of Actinobacteria in nature is the carbon cycle. What they do is they help break down organic materials, which is important for returning nutrients to the soil. The process supports plant growth and maintains healthy ecosystems. Now, some Actinobacteria have unique structures called filamentous hype, which look a lot like the branching structures found in fungi. This similarity has caused some confusion among scientists, leading to the naming of certain bacteria like Mycobacterium with myco, MYCO, in their name, suggesting they're fungi when they're actually bacteria. Now, in the human body, many Actinobacteria are harmless and even beneficial, living in our microbiomes. However, like many bacteria, some species in this class can be harmful and cause infections. So understanding both the beneficial and pathogenic roles of Actinobacteria is important for you guys as ST students, especially when considering infection control and the use of antibiotics doing a lot of the surgical procedures. Now, we get into actinomyces. This consists of bacteria that are gram-positive and can thrive in low oxygen environments or can survive with or without oxygen. These bacteria are filamentous rods, meaning they have long thread-like shapes and they do not form spores. Actinomycetes bacteria are commonly found in the mouth, the nose, and the throat of both humans and animals. When they grow in the lab, they can resemble dust bunnies because of their filamentous appearance, which can sometimes lead to them being mistakenly identified as fungi. Now, we get into some diseases. Actinomyces bovis is a bacterium that causes actinomyces in cattle, commonly known as lumpy jaw. This condition results in the formation of pus and destruction of the jawbone and soft tissues in affected cattle. While the name actinomyces may suggest a fungal infection, it's actually caused by bacteria. Now, in both animals and humans, actinomyces infections usually arise from the normal presence of actinomyces in the mouth, the nose, and the throat. As a result, these infections typically appear in the face and the neck areas. The bacterial lesions lead to chronic swelling, the formation of abscesses, and the creation of sinuses or granulomas. Infections can penetrate multiple layers of tissue, heal, and then become reinfected multiple times. In some rare cases, actinomyces can affect other parts of the body, such as the chest, the abdomen, the pelvis, and even the brain. These severe infections may require prolonged antibiotic treatment. Now, in figure 14.2, you have a facial fistula of the left cheek due to none other than actinomyces or mycetomoa, which is an infection caused by the gram-positive fungus-like aerobic bacteria of the order of Actinomycetes, which is a slow, progressive, destructive infection of the cutaneous and subcutaneous tissue, as you can see in the image, and can progress, unfortunately, all the way to the bone. We have to also understand Corynebacterium. This includes a group of bacteria that are non-spore forming gram-positive and typically have a club-shaped or a V-shaped appearance. These bacteria can thrive in oxygen-rich environments, which is aerobic, or in low oxygen conditions like facultative anaerobic, and they are generally non-motile, meaning they're not moving a lot. Okay? They produce lactic acid by fermenting carbohydrates. They are catalyst positive, which means they can break down hydrogen peroxide. There are two main groups. You have Corynebacterium differia, which is known to cause diphtheria, and a variety of other species referred to as differithroids. Now, Corynebacterium is a pleomorphic bacillus, meaning it can take on a lot of different shapes. Think of it as like a shapeshifter. Now, when stained with methylene blue, it shows granules, but this characteristic isn't unique to this species, so it can be solely relied upon for identification. Now, after the cell divides, it exhibits a unique jerking motion that arranges the cells in patterns that kind of look like Chinese letters, which can help in its identification underneath the microscope. There are three main strains of Corynebacterium differia. We have gravis, intermedius, and metis. All three strains can produce a very harmful substance called diphtheria toxin, and they typically live in the upper respiratory tract. So how does this strain differ? Well, we look at the virulence. Now, this term refers to how harmful a bacteria is. The difference in how dangerous each strain is come from how quickly they grow and how much toxin they actually produce. The faster a strain grows, the quicker it can take in local iron from the environment, which helps it to actually produce more diphtheria toxin at a faster rate. So we start with gravis, which is actually the image in the middle. This strain is the most virulent, meaning it's the most harmful, and has a generation time, which is the time it takes to grow and divide, of 60 minutes. This means it can grow rapidly and produce a lot of toxin quickly. The next one is intermedius. This strain has a longer generation time of 100 minutes, making it a little less virulent than gravis, but it still poses a risk. And the last one is medius, which you see on the left. This strain has the longest generation time at 180 minutes, making it the least virulent of the three. Now, understanding these strains is important for us. Why? Because recognizing the potential severity of infections caused by this particular Corynebacterium, especially the gravis strain, helps in assessing patient risk. We're able to know that the bacteria can produce toxins quickly, informing infection control practices where we can emphasize the need for prompt treatment. If a patient is infected with one of these strains, we have to be prepared to follow specific protocols to minimize the risk during surgical procedures. Now, we look at cutaneous diphtheria. This is less common and spreads through direct contact with skin lesions from an infected person. If the skin is damaged, like through an abrasion or a cut, the bacteria can enter deeper in the body. So the infection usually starts as a small raised bump or papule that can develop into an actual ulcer. The ulcer is marked by a grayish membrane, which is a sign of the infection. So in figure 14.4, which you see in front of you, is a skin lesion on the leg. But the chapter also tells us that you can also have respiratory diphtheria. This is more common and primarily spreads through droplets when an infected person coughs or sneezes. This makes it easier to catch in crowded or close contact situations. It can affect different parts of the throat. So it can affect the front part of the nasal passages. It can get into your pharyngeal and your tonsillar or your tonsils. So that's involving the throat, the tonsils, where now you have a sore throat, difficulty swallowing, your larynx, your voice box. It can affect that, causing hoarseness and breathing difficulties. We have Corynebacterium diphtheria invading the respiratory tract, producing toxins that causes symptoms like fever, sore throat, weakness, and swollen lymph nodes in the neck, leading to a condition called bull neck. So the lesion forms that injures epithelial cells, which are the cells lining the respiratory tract. Your blood plasma leaks into the area, creating a fiber network mixed with bacteria, dead cells, and immune cells. Within a few days, a very thick gray pseudomembrane develops that adheres firmly to the tissue, and then now breathing is obstructed. Removing this membrane can cause bleeding, think about it, because of how it's attached from underlying tissues. So the key is the production of the toxin. The toxin is encoded by a gene introduced by a lysogenic bacteriophage, which is a virus that infects bacteria. When a host is iron deficient, the bacteria can produce large amounts of toxin rapidly. Early treatment are also administered to just eliminate the bacteria. Patients may require some type of airway treatment just to keep their airway open. Infected patients are kept in isolation on strict bed rest to prevent spreading the infection, and those who recover will cough up and clear the dislodged membrane. So in figure 14.5, it's just a child presented with characteristic swollen neck, sometimes referred to as bull neck, that we just talked about. Now, the family of Mycobacteriaceae is part of the class Actinobacteria and is known for its distinct characteristics that set it apart from the other bacteria. So there's a unique cell wall structure, It has a cell wall rich in lipids, especially waxes, which include mycolic acids. This is just a type of fatty acid that is unique in structure and is why these bacteria do not consistently stain purple or red when using the gram stain technique, making them gram variable. To identify mycobacteria, a special staining method called acid-fast staining is used. So when stained correctly, cured correctly, they appear red against a blue or green background under a microscope. This method is particularly important for diagnosing diseases like tuberculosis. Now, under the microscope, Mycobacteriaceae bacteria may be straight or slightly curved. They can also have filaments or branches extending from their cells. They are non-motile, meaning that they do not move on their own and they do not form spores. These bacteria are aerobic, which means they need oxygen to grow. They can thrive in a lot of different environments, including areas where there is plenty of oxygen. So a couple diseases. Leprosy is caused by Mycobacterium leprae, leading to skin lesions and nerve damage. We mentioned tuberculosis, caused by Mycobacterium tuberculosis. It's primarily affecting the lungs, but can also spread to other parts of the body that lead to extremely serious health issues. There's also non-TB respiratory infections, which is just another species in the family that can cause infections in the lungs that are not caused by Mycobacterium tuberculosis. Now, it's important for us to understand this disease because we have to know the transmission routes of TB and leprosy, which help in early detection and intervention. Because TB is airborne, we have to be well versed in infection control measures just to protect ourselves and other patients, especially in the operating room where respiratory infections can pose a significant risk. Also, I want you to think about proper aseptic technique, especially when working with these patients who may have infections caused by this Mycobacteriaceae, just to minimize the risk of cross-contaminating an infection. Going a little bit further into Mycobacterium tuberculosis. Okay, so it's a rod-shaped bacterium and an obligate aerobe, meaning again, it needs oxygen to grow. It's known for its waxy cell wall, which contains mycolic acids. It has a unique structure that slows down its growth and provides protection against enzymes produced by immune cells that they try to destroy. So the bacterium prefers areas with a lot of oxygen concentration, and that's why it often infects the upper lobes of the lungs. But it has a slow doubling time of 20 hours compared to only 20 minutes for bacteria like E. coli. So keep in mind that TB is a highly contagious disease that spreads from person to person through respiratory droplets. These droplets can become aerolized when an infected person talks, coughs, sneezes, which allows the bacteria to be inhaled by individuals nearby. In healthcare settings, right, airborne precautions are going to be important because we want to minimize exposure. Patients with active TB are often placed in negative pressure rooms with filtered air to prevent the spread of bacteria to other areas. The chapter talks about classes of tuberculosis. We have LTBI, which is latent TB infection. This occurs when a person has been exposed to TB bacteria and inhaled them but shows no symptoms of active disease. Individuals with latent TB are not contagious to others. The bacteria can remain dormant in the body. If a person's immune system does become weak, latent TB can progress to active TB disease. And then there's TB disease. This is the active form of the infection where symptoms developed and the individual is contagious. Symptoms may include a persistent cough, weight loss, fever, and night sweats. There are two paths for tuberculosis: primary TB and reinfection. So, the initial stage of TB infection, known as primary tuberculosis, begins when the bacteria enters the lungs and causes lesions. This stage leads to an acute exudative phase where the bacteria rapidly spreads through the lymphatic system. The resultant lesions often form in the lower lobes of the lungs. These primary lesions can heal over time, but may leave behind some kind of scarring in the lungs. And then there's reinfection tuberculosis that occurs when the bacteria reactivate after surviving the primary infection, typically characterized by the formation of new lesions. Now, these lesions tend to occur in the apex or the top of the lungs and can spread downward into the lower lobes. This chronic form of TB often results in lesions that secrete exudative fluid, leading to unfortunate further complications. So we look at the types of lung lesions. You have exudative lesions, where these are caused by the host's inflammatory response and contain kind of liquid. The lesions can progress, leading to necrosis, which is death of lung tissue, healing, or the formation of a tubercle. And then you have productive lesions. Productive lesions do not contain any type of exudate. Instead, they form a structure called granuloma around the bacilli, which is the TB bacteria. Now, a granuloma consists of a dense collection of immune cells surrounded by lymphocytes, monocytes, and fibroblasts. The center of the granuloma may become calcified over time, so it gets kind of hard. The outer part of the granuloma develops a fibrous capsule known as a gnome complex, which can remain dormant for many years, or it can actually eventually die. So figure 14.7 is a x-ray showing advanced tuberculosis, and I want you to look at the presence of bilateral pulmonary infiltrates, and then you can kind of see like a little bit of caving formation in the right apical region. Another thing the chapter talks about is the impact of COVID-19. Statistics from 2020 show an unexpected decrease in reported TB cases, which raised concerns like what's going on. Now, the COVID-19 pandemic may have led to TB infections being overlooked or misdiagnosed as public health focus shifted predominantly to COVID-19. Many individuals with early symptoms of TB may have been reluctant to seek health care due to fear of actually contracting COVID-19, potentially leading to delayed diagnosis and treatment. So for us, understanding the stages and types of TB lesions is going to be important to be able to recognize potential symptoms in patients and, again, ensure proper precautions are taken. When a person is exposed to Mycobacterium tuberculosis, the immune system reacts by activating macrophages, which is a type of immune cell. This reaction can be detected through a skin test. Now, the TB skin test uses purified protein derivative, or PPD, which is an extract from cultures of Tuberculobacillus. When injected into the skin, it can trigger a hypersensitivity reaction in individuals who have been exposed to the bacteria. So the Mantox test is the most widely used and accurate skin test for TB. However, when we think about how it works, okay, you have a small amount of PPD injected just beneath the skin into the forearm. After about 48 to 72 hours, the injection site is assessed for reaction. A raised reddened area at the injection site indicates a positive reaction, suggesting that the individual has been exposed to tuberculosis. So we look at hypersensitivity, right? This refers to the immune system's exaggerated response to an antigen. And of course, in this case, we're talking about the PPD, which leads to the characteristic swelling, and then you see that redness. The Mantox test is that specific test used to detect TB exposure through the administration of that PPD. We also have to look at drug-resistant tuberculosis. Now, Mycobacterium tuberculosis can develop resistance to standard anti-TB medications. The primary reason for this resistance is the misuse or mismanagement of drug treatment. Multi-drug resistant TB, or MDR TB, is caused by strains of TB that have mutated and become resistant to the two most effective TB drugs, isoniazid and rifampin. This makes treatment just that much more complicated and requires a longer duration with different medications. We also look at extensively drug-resistant TB, which is XDR TB, is an even rarer form that is resistant not only to isoniazid and rifampin, but also a fluoroquinolone and at least one of the 3 injectable second-line drugs, which means it is significantly limited with treatment options and poses a higher risk to the public health. So then this brings up Mycobacterium leprae and leprosy. Now, Mycobacterium leprae is closely related to tuberculosis, believe it or not, because they share the same unique waxy cell wall structure that requires acid-fast staining for identification. It grows best at slightly lower temperatures than body temperature, which explains why it typically affects cooler areas of the body like the fingers, the toes, the ears, as well as the nose. Hansen's disease, also known as leprosy, is a chronic infectious disease that has been feared throughout history. Individuals with leprosy face so many social stigma and isolation. M. leprae is most likely transmitted in a manner similar to TB and affects both the skin and the peripheral nervous system. It can result in severe, disfiguring skin rashes and loss of sensation in affected areas. We look at 2 forms of leprosy. We have tuberculoid leprosy, characterized by a strong immune response, fewer lesions, and localized skin systems. And then you have lepromatous leprosy. It involves a weaker immune response, more widespread lesions, and significant nerve damage, leading to loss of sensation and deformities. Now, with tuberculoid leprosy, the immune system responds strongly with a cell-mediated immune response, meaning that the body uses its own immune cells to actively fight the Mycobacterium leprae infection. Patients typically develop only a few skin lesions. Now, these lesions may not cause significant damage, but individuals often experience a complete loss of sensation in the infected area because the bacteria damage peripheral nerves. With lepromatous leprosy, if a person's immune system cannot control the infection, it can progress to lepromatous leprosy. In this form, there are high numbers of M. leprae cells, making it highly contagious. Patients develop extensive skin damage characterized by flat or raised lesions across the body, leading to significant disfigurement. So facial features may become thickened, often described as lion-like. You have a hallmark of lepromatous leprosy. The nose may flatten and collapse due to the destruction of cartilage, and there may be even loss of eyebrows. In severe cases, bones and cartilage can be damaged to the extent that amputation of digits is necessary. Figure 1411, you have democratic skin plaque with elevated borders and a loss of sensitivity in the central region due to the tubercular form of leprosy, or Hansen's disease, HD, caused by the bacterium. Okay, now Mycobacterium leprae and you have the characterized by that presence of the asymptomatically distributed solarity of the skin lesion. Basically, you can kind of see the raised area, everything that we had just talked about. Okay. And keep in mind that there is a loss of sensation. Couple more pictures of Mycobacterium. In figure 14.3, you have lepromatous leprosy, and you look at different standards, right? Classified as the Mycobacterium, right? Where you look at that saddle nose deformity. And this is due to the loss of that nasal cartilage. And you also notice that in that particular image in 14.13, there is the lack of eyebrows. And in figure 14.2, you have hands of an individual, again with leprosy, also known as HD, that Hansen's disease. And in this image, you see how the peripheral nerves have been affected and the severe degeneration and mutilation of all of the fingers. Okay, so it looks like amputation may have, have been something that had to occur. So you kind of just look at the different severities of the disease. So how does leprosy damage the human body? Well, let's take a look at this video.
Leprosy brings to mind images of biblical beggars and lonely leper colonies, and seems for some to have all but disappeared into the history books. But leprosy still haunts many parts of the world, and there's an absurd amount that we don't know about the disease. Leprosy has been around since at least 2000 BC, but after all this time, we still don't really know how the disease is transmitted. We don't know where it starts in the body. Oh, and we can't grow leprosy bacteria in a culture. But maybe we should start with what we do know.
My name is Cressida Madigan. I'm an assistant professor at UC San Diego in the molecular biology section, and I work on leprosy and other neurological infections to try to understand how these infectious agents are damaging our nervous system during infection. I love Mycobacteria, which is the kind of bacteria that causes leprosy. They're very unusual organisms.
And part of what makes leprosy bacteria, or Mycobacterium leprae, so unusual is that it is thought to be only able to replicate inside a living cell.
These bacteria can't be grown in a lab the way that we grow other bacteria. We don't know why. We don't know if maybe we're just missing a key component of the recipe that it needs to grow. Could also be that leprosy simply cannot grow outside of an animal.
So scientists like Dr. Madigan study leprosy inside living animals like mice, armadillos, and zebrafish. But this still makes the bacteria difficult to observe and run tests on, especially since it occupies a unique part of the body, the nerves. It's the bacteria invading the nerves and the immune system's response that eventually causes the major symptoms of the disease. So while we aren't really sure how the disease gets into the body or how it travels to the nerve cells, we know it infects what are called Schwann cells, which produce the protective myelin sheath that covers neuronal axons, helping to conduct electrical impulses along the nerves.
So there's some evidence that when M. leprae infects Schwann cells, it stops those cells from being able to make myelin, which is this sort of insulating wrapping that surrounds the axons of our nerves.
Something about the infection causes the protective myelin to die. And this is because the bacteria is destroying the small but mighty mitochondria in the nerve cells.
So mitochondria are sort of like the batteries within cells. They produce the energy for the cell so that it can function. So as a cell, if you don't have mitochondria, you will essentially die at some point, right?
Over time, the nerve axons also become damaged by the immune system's response to the bacteria, which is why people with leprosy can lose feeling in their skin. But the immune system isn't activated immediately, and that's because leprosy bacteria is notorious for moving at a snail's pace. While other bacteria like E. coli can replicate in as short of a time as 20 minutes and disseminate quickly, leprosy bacteria replicates only once every 2 weeks or so. So someone could be infected for years and not experience any symptoms because the bacteria isn't setting off any alarms. This also makes the bacteria difficult to treat.
Many antibiotics work by targeting bacteria that are replicating. So if you're a bacterium and you're not replicating, then you will be naturally resistant to several classes of antibiotics.
To treat the disease, you'd have to take a cocktail of various antibiotics daily for up to 2 years to make sure the drugs hit the bacteria at the exact moment it's trying to replicate. And you would need to take multiple antibiotics at one time to ensure the bacteria doesn't become resistant to the treatment, which is not ideal. But the good news is, leprosy really isn't that contagious compared to other diseases.
Part of the stigma of leprosy is that this disease is very contagious, but that's actually not true.
In fact, you need to be around someone with leprosy for a long time to catch the disease. It also helps that 95% of people are naturally immune to leprosy.
So in order to contract leprosy, you have to be living with a family member who has the disease, who is infectious, and you have to have close, intimate contact with that person for a period of years.
And while the symptoms are undesirable, leprosy doesn't directly cause death. In some patients, the symptoms take decades to manifest. So, like we said earlier about the leprosy bacteria,
they're very unusual organisms. The Byzantines knew about leprosy. They knew that it was infectious. And so to try to stop the spread of leprosy throughout the Byzantine Empire, the Byzantines would assign leprosy patients into leprosaria, which were like very posh leper colonies, essentially. The leprosaria were actually really nice places for patients to live. They got free medical care, free food, housing. And so life was so good within the leprosaria that people started trying to fake the symptoms of leprosy in order to be admitted.
Oh, to be a leper in the Byzantine era. Am I right?
All right. Now, we also have Nocardia and Nocardiaceae. Nocardia species are a type of aerobic bacteria found in environments like soil, decaying organic matter, and water. They belong to a group of bacteria known as actinomites. We also look at Nocardias. This is an uncommon infection caused by these Nocardia bacteria. It's really important to just mention that this infection can occur in both humans and animals. About 80% of cases of Nocardias manifest as some type of pulmonary infection in the lungs, disseminated infections, meaning that it just spreads throughout the body, brain abscesses, yes, you've heard that correctly, localized infection in the brain, and the remaining 20% of the cases may present as, let's say, like cellulitis, which is inflammation of the cells or skin infection. Infections that can involve other organs such as like your kidneys, your joints, your heart, your eyes, and even your bones. Now, patients with nocardias may experience different symptoms depending on the air, right? So like if we talk about pulmonary symptoms, you're coughing, you're having difficulty breathing, you're having chest pain. With the central nervous system, you think about headaches, lethargy, which is just extreme fatigue. There may be confusion, some seizures, or sudden neurological deficits, which is just like a sudden loss of function. And the very last that we're going to talk about is streptomyces. These are a group of non-motile filamentous gram-positive bacteria that belong to the class of Actinobacteria. They are typically, again, found in soil worldwide and play a big role in soil ecology. These bacteria produce spores from special structures called sporophores. They are known for the distinct smell of wet dirt, which comes from chemicals called geomines that they actually release. Streptocytes can break down a variety of organic materia, including sugars and amino acids. Streptocytes are important because they produce more than 50 different antibiotics and other medicinal compounds. We look at streptomycin, used for tuberculosis, neomycin, which is a topical antibiotic, chlorophyll, used for serious infections, tetracyclines, which are a broad spectrum antibiotics, and ivermectin, which is used for parasitic infections. Now, while streptomyces are beneficial, they also can cause infection with common clinical signs being myosoma. This is a chronic localized skin infection that has granuloma formation, which is just a collection of immune cells, and pus formation as well as inflammation. So, understanding this can cause infections is going to be important for us, especially when dealing with patients who may have been exposed to soil or have an open wound. Now, if you have any questions, concerns, please make sure that you reach out. Let's look at our objectives again. Okay, so what do we talk about this evening? We defined key terms, we compared and contrasted the different genre of Actinobacteria, we described Corynebacterium pathogens and the diseases that they actually cause. We described the Mycobacterium pathogens and the diseases they cause. We looked at implications of drug resistance in tuberculosis, and we had to kind of think critically in relating all the information that we covered and how we in the operating room have to be aware of the care on a broader global community. Again, this is your mini session for ST210, Week 6, Chapter 14, Actinobacteria. On that, I look forward to seeing everyone who can attend the live session.